Stay nice and dry? Excellent. Welcome to Mosses and Lichens. Today we have Dr. Mack, Frantz, and Sue Studlar is with us. She's also a doctor, correct?
That's correct.
Correct. I don't know you, Sue, and I apologize for that. Anyway, they're going to be bringing us lots of really good information on mosses and lichens. Big welcome.
Good morning, everyone. Thank you all for being here. Like Keith said, my name is Mack Frantz. I'm the zoologist based out of the Elkins office with the DNR. And my role is to manage occurrences of non-game species in the state, which then you got to ask, what does it have to do with lichens? Well, I was very fortunate the year Dr. Sue Studlar retired from WVU that I took a special topics course on lichens, where I went through the WVU herbarium collection for bryophytes and lichens, and used that to come up with a field guide, a user-friendly field guide for lichens. And because of Sue, I got hooked on lichens. I consider myself an amateur hobbyist. I'm no expert by any means, but lucky for us, Sue is the resident expert, at least much more so than me. So if there's something I don't know, she surely could pitch in. Anything you wanted to add, Sue, or you want to introduce yourself later?
Oh, why don't I do that later?
Okay, so today we're gonna start with lichens. We're gonna try to have a little break between the lichens and mosses, and so we'll be here in the morning, and then in the afternoon we'll still meet in the main foyer of the lodge, and then depending on the rain we'll decide whether to go out if it's not raining too badly, or if we see there might be a break in the rain, we can come back here and look at the bryophyte lichen specimens that we collected and just do some more stuff indoors. So that's also a potential option. Today we'll be discussing the macro lichens, which are essentially the ones that you can easily see without strong magnification. We will cover some of the micro lichens, your crustose lichens, some of the more recognizable species. What exactly are lichens? You've probably received a couple different definitions of them, or have somewhat of an idea. But they are actually a composite organism. Can everyone hear me okay?
No.
No.
No, but Steve went to get you a portable mic. Mac, it's really good to use. You know, we're
Okay.
all older people who don't hear that well.
Okay. Whenever the mic comes, I'll try to use that in the meanwhile. Okay.
Speak for yourself. All older people that can't hear you?
Well, many of us.
Even older people who can't
And I
hear.
can fall into that category, however you might have offended.
You sure don't?
Yeah.
Might still not be any better. I'm the only one up there. Yeah, I know what you mean. See that much. Yeah. I don't know one thing about it. Whatever I learned.
That's the first thing. It's a combination of
I do feel that.
That's the first thing. That's the only thing.
Yeah. And I know British songs.
Does this mic help a little bit?
Yeah.
Okay. This is going to be a little bit distracting to me, but I'll try to get used to it. So like I said, lichens are a composite organism. The definition you typically heard is that they consist of a algal or photobiont partner, or a cyanobacterium, which has also been called blue-green algae. They are both photobiont partners, meaning that they make their own food from energy from the sun. And lichens also consist of the fungus, and the fungus has the most influential role on how the final lichen structure looks. Historically, they've always been kind of given as a textbook definition of mutualism, that they both benefit from the relationship, but we'll see that that's not exactly the case. It is more of a controlled parasitism, because the algal or the algae or cyanobacterium is actually placed within a layer of the lichen, and the fungus utilizes that to derive benefits from the algae. So it's actually more of a controlled parasitism. The other way I'd like to look at lichens is that it's a fungus that learned how to farm the algae. But they both do get benefits from it. The algal or cyanobacterial partner provides food, vitamins, and amino acids. It's actually the cyanobacterium that can remove nitrogen from the air and use that in order to make amino acids. The fungal partner does provide benefits to the algae in the form of protection. It shelters the algae from the sun, and it also, there are some chemical compounds that the fungus produces, anti-herbivory compounds, among other kinds of compounds that can protect the algal partner. And as a result, the algae can exist in environments that it typically wouldn't be able to because it receives that protection from the fungus. So there is an expanded habitat. For instance, the most common algal partner is Sharboxia, and it has been found statistically more in areas of boulders than it would be able to do by itself from this beneficial relationship. However, surprise, there's always, the more you look into something, the more you realize you don't know. There actually might be a hidden third partner within lichens that has you might have read about in the news. And it actually consists of a Basiomycetes fungus. The fungus for lichens is usually the Ascomycetes, your cup fungus, but in this case, the third partner actually might be yeast. There's been a lot of studies done where genetically lichens can be genetically similar yet look totally different, and they wondered why. And it turns out it might actually be because of that yeast. And it also produces some secondary metabolites that provide some different kinds of defense compounds. Depicted here on the right is the wolf lichen, which that yeast produces that yellow pigment. And you can think of yellow pigment in lichens as a sort of sunscreen, a sunscreen that protects it from too much sun radiation. Why are we talking about mosses and lichens in the same course? Well, they are informally grouped together in a group called cryptogams. And cryptogams means secret or hidden marriage, which means that they both produce by spores. They don't have any kinds of seeds. As far as classification for lichens, like I mentioned earlier, it's the fungus that has the most influence on the final composite form. Both the fungal and algal partners have their own scientific names, but when it comes to the lichen itself, it's known by the name of the fungus. The majority of lichenized fungi are part of the Ascomycetes, the cup fungi, which if you go on the outing today, we'll actually get to see what that form looks like when it's not a lichen. Otherwise, there's only about 20 lichen species out of the 18,000 or so that are with the Basidiomycetes as the major partner, and that's more in your tropical areas. There's about 40 genera of algae and cyanobacteria that are capable of forming these relationships with the fungus and with the right environments. And believe it or not, lichens only within the last 20 years have received common names in order to make it more accessible to people. And it's the work of Brodo and many other, the Sherinos and other collaborators that have made common names for the lichens. And you can see those in the tome Lichens of North America, which we will discuss resources here in a little bit. Lichens are considered ecological indicators, and ecological indicators are measurable characteristics of structure, composition, and function, with that consisting of an ecosystem, all your biotic and abiotic factors and those interactions. As such, lichens have been used for air pollution monitoring. They also are known for bioaccumulating contaminants. You can use that as a relative index. And they can also be used as indicators of climate change because of the changes in the availability of water and also because of temperature changes. The U.S. Forest Service considers them ecological indicators. They're within the Forest Inventory and Analysis Program. And the Lichens Community Indicator Index, what they do is they collect samples of lichens to be able to identify all the species, and then from there they determine the abundance of each of those lichen species. And then over time, through a trend analysis, they can see how those community structure changes because potentially of climate change and air pollution, among other things. And so here's an example of the abundance of a particular lichen species, and potentially over time, you could see a shift to higher elevations as the climate changes. Lichens have been used for monitoring in West Virginia as well, within the Mon National Forest. There was a study done in 1987 with a follow-up study in 1992, just to kind of see how air pollution might have changed at Otter Creek and Dolly Sods. Air pollution during that time remained low, but they did see an increase in certain heavy metals. They also found higher sulfur, nitrogen at Dolly Sods, and as you would expect, you did see a correlation between elevation and the amount of acidic precipitation present, where that's coming from out in the Midwest, and then once it reaches our mountains, then that's where it deposits. So lichens, there are different habitat types. That's one way to try to indicate what kind of lichens you can find in an area. Lichens need water, air, nutrients, lights, and substrates, the substrates being what they attach to. Habitat consists of biotic and abiotic factors, both physical, chemical, and biological. And for our purposes today, habitat is just the area in which a lichen lives. And definition of lichens being ecological indicators of habitat is that a species is found more commonly inside the habitat than outside of it. So in a anthropogenic area or open exposed field like that, you'll expect certain lichen growth that is very tolerant of a lot of sun, which all lichens do need, sun exposure. But there are also shade-tolerant lichens that you would find, such as cathedral state forest, that you wouldn't find in that environment. So the Forest Service, like I said, they actually consider lichens habitat indicators as well. They have one example of a species in their environmental impact statement. Essentially, if any kind of work done on the forest, you need to abide to federal law for NEPA. How is that going to affect the environment? And one of them that's an indicator of habitat is the sand-loving Iceland lichen, which it needs for at least a good majority of its life cycle mature forest. And it mentions in there how the potential of management with fire maintaining oak forests, how that might affect it. As far as resources go, for use in the classroom today, you have a guide in front of you. I've been told that can only be used in the classroom or I'll get in trouble, but you can find that guide for free online. And that is, it's a little bit different from our guides you can find out there because it's based on habitat rather than keying it out by growth forms and that sort of thing first. Yes. Otherwise, I'm the one that gets in trouble. So that's what I've been told by the others. There are some other great local resources. I'm part of the OMLA, the Ohio Moss and Lichen Association. They have forays each year and some annual meetings. Membership is relatively cheap too, but it's a really great group of experts on all things mosses and lichens. I got involved because of Sue's recommendation. There's also the book that I mentioned earlier called Lichens of North America, which I was outside with it, and I don't recommend taking this book outside. But it does have a dichotomous key in it, and it also has a lot of pictures too, when you're kind of starting out and kind of get an idea of growth structure and form. There's also an additional book to that I highly recommend once you get away from the pictures and you just want to rely on the guide. The Keys to Lichens of North America has many more species in here that you can key out that the book cannot. And also has some of the name changes in there too. So this is, I recommend to get this if you also get the Lichens of North America book.
Mack, is there a way we can get a digital copy of this? Email that, so can we email you or something like that?
Yes, yeah, I can definitely do that.
Okay. Thank you.
There's also another book that's nice, that it's called Common Lichens of Northeastern North America by McMullen and Anderson, which is my understanding. Troy was one of Sue's students, I believe. Or no, okay. Okay. She wishes she could claim them, but it is organized in here by growth forms of lichens, which is kind of nice and handy.
What was the title of that again?
Common Lichens of Northeastern North America. Another one is there's a free Ohio field guide, which Sue brought for you guys. If you want a copy of this is a really excellent resource given out by the Ohio DNR. So there's a bunch of these available up here that you can take one today with you. If you decide to really get into the weeds, stuff that's over my head, there's a book by Nash called Lichen Biology, and this really gets into all the nitty-gritty stuff that you find in the research articles.
Who's that? Nash?
Nash. Yep. It's a little bit on the pricey side, but if you really want to get into the weeds, that's a great book. There's also an ongoing effort online, a digital consortium of North American lichen herbaria, where the WVU's lichen collection is digitally archived, and you can find those collections online through that consortium, and there's hundreds of photos from lichen collections around the United States. It is a really excellent resource that I go to all the time, especially when I run into something with a lichen I'm trying to key out that doesn't necessarily fit within the resources that I'm reading. It's a really nice, up-to-date resource. And then finally, I don't know, you can always contact the DNR and see if we have any left. The last I heard, we don't have too many. It's out of print, but the Macro Lichens of West Virginia by Don Flanagan is a really excellent resource. And I know if you're part of Oman, they have a lot of copies of it left because Don Flanagan was really involved with them, and I believe native to Ohio, and he's made some lichen guides for Ohio. So this is another one that I recommend to get your hands on. As far as equipment goes, a hand lens is essential to be able to see the reproductive structures on lichens, texture, and among other things. So a little hand lens. If you, for some features, it's easier to see underneath a dissecting scope. That's recommended. For photography, if you don't have money for a nice digital camera like me, then you can go to Amazon and just buy a $10 macro lens, and that seems to take pretty good pictures of lichens. Another thing you might want to take out in the field is a water spray bottle, because the color of lichens can significantly change once they become active. The algal or cyanobacterial partner will become more prominent whenever the species is active and actually producing food and it's not dormant. And then For a lot of lichens, you need to be able to determine what kind of acidic substances or compounds are in it or that it produces, and so you have to do some chemical spot tests. Luckily, a lot of them, though, are easily accessible, and at least if you can't get some of the chemical compounds, it can help you narrow it down to at least a few species. This is an example here of a dry common toad skin versus a wet one. And this one, the one on the wet, is just in the process. It's a transitioning into being fully active. It is not bright green just yet. But that whenever a lichen is dry and brittle, that does not mean it's dead. It is just dormant. And once it's wet, the water runs over it, the foliose lichen, then that's when the photobiont partners become active once again.
So how long does that process take?
It really doesn't take too long at all. You can just spray it out in the field a lot of times, wait a few minutes, and you can see the difference. Here's an example here of a dogtooth lichen where with Peltagira is the genus, and they have a cyanobacterial partner, and so usually when it's a cyanobacteria, it's a darker color. It's like a dark brown, dark black, or a very dark green, and that can kind of help it. That it'll look a lot different when it's drier. The very first thing you want to look at when you're IDing lichens are the growth forms. So the vegetative body of the lichen is called the thallus. So you're usually trying to key out the thallus along with the reproductive structures. Typical growth forms, they're put in these columns, but you'll find there's a lot of hybrid forms. Foliose lichens, fruticose, which are kind of more pendulous or tufted. You have your squamulose, which are in between being macrolichens and microlichens. It's somewhere in between there. And then you have your crustose lichens, which are as thin as a layer of paint and usually embedded in the substrate. So you cannot remove it unless you actually remove the substrate with it. And that's the majority of lichens. However, crustose lichens are out of the scope of what we can cover today. But there are some great workshops out there that you can go for crustose lichens. But we will cover some of the ones that you will more commonly recognize. And note, like I said, there are a lot of hybrid growth forms, especially with crustose, fruticose hybrid forms. This is the a cross section of a typical foliose thallus. You have the upper cortex, which you can kind of think of as the skin of the lichen. So it protects the algae or cyanobacteria from receiving too much light. It provides the shelter. The next layer is a very thin layer where the fungus stores or keeps the fungus for deriving benefits from it. The bigger structure is the medulla, which is right underneath that green layer there. It's a loose fungal hyphae. That's where water is stored or moved. Some chemical compounds are made, and foods move there. And so that's kind of a more loose, bigger region. And then you have the lower cortex of it, which difference between the upper and lower skin makes a difference in lichen identification. And you also have risings, which you can think of as almost roots for the lichen, which helps attach it to the substrate, which just the foliose lichens have these type of typical growth forms from the thallus. You'll see for some of the other lichens we covered that they have different means of attachment. We also have your apothecium or apothecia. You'll notice I'll say it in some different ways, which produces the asexual spores for the lichen, which is one of the reproductive structures. So water splashes on the cup, and that ejects some of the spores, and then it's hopefully carried away by the water, air, or an animal, and to form a new lichen elsewhere with the the right fungus and algae meet together in the right habitat.
Is there a sexual phase?
Actually, those spores would be sexual. Those are produced by meiosis. So it would be the
Right.
So they would really have to be called sexual, but for the fungus only.
Right.
Yeah.
So yeah, listen to Sue on that. But yeah, so that's the roundabout way that they do sexual reproduction. That's correct. And they can't do it themselves per se, but they have to. That's the roundabout means that they
can't
do
do
it.
it together.
Yeah.
That's the problem.
Yeah. As far as the risings go, they come in many different forms. They can be simple, so just a simple straight rising. Placement of the risings matter. You might have in some keys whether you have a bare margin on the other side of it. Some of them can be tufted with both little and large risings together. The color of it, some of them can be branched, some of them can be at right angles. And you also have tufted ones, like in the form of Peldagira, that kind of looks more like roots or threaded. To go over the first growth form, foliose lichens, they're very leaf-like. They lay flat, and they vary from being loosely to tightly attached. And when it comes to ID and amount, the upper and lower thallus surface, that they can differ in color. Some are just jet black underback, some are white, and some of them maybe it looks about the same. The edge of the thallus, where it's smooth, ruffled, or whiskered, helps. So some lichens produce these cilia-like projections in the ruffle lichens, and you can see on here this hammered structure of the Parmelia saccata. You have some fringe wrinkle lichens that kind of look really lacerated like that. So those are kind of some of the features to look for. The air-growth form is fruticose, which those symbols up there kind of follow the same in the book. Just that's at your desk, just to kind of get an idea. They're shrubby, tree-like, or hair-like. They typically do not have a distinct upper and lower surface like your foliose lichens do. And they're weakly attached to the substrate. They don't have the risings that you see earlier. And sometimes you need to do a cross-section of them, see if they're hollow, solid, do they have a elastic band in it, cylindrical, flattened, branched, or unbranched. Your squamulose lichens, those are the ones that are somewhere between your macro and micro lichen. They're these little pebble-like units, little scales called squamules. They're also referred to as sod flakes, just because you find it typically within a sod on top of the soil. Some are strictly squamulose, consisting of those little pebble cortex units, while some have a secondary fruticose structure, which are these little stalks here, your podia. And a lot of times with your squamulose lichens, they're very much like your bryophytes, where they live in these mosaics with multiple species present. So I have an example here of some squamulose lichens where there's 4 different species present within this little area, if you want to pass that around and kind of take a look. On the other side of the room too, I'm going to go ahead and start passing around lung lichen, which is Loberia pulmonaria, and this is an excellent indicator of mature forest. I only seem to find this in places like Gardenier Knob, Blackwater Falls, and it's also, there's some of it on the trail today too, if we get to go out there. But you can see it's has a reticulate ridge throughout the thallus. Kind of looks like a dried peanut shell kind of structure. And we'll be talking about some of the reproductive structure shortly. One of which is our cydia, which are kind of like, you'll see they kind of look like mini fingers or hot dogs. And those are on the margins of the lichen thallus, and so that's called isidiate. And the top of the lichen is also very fuzzy or tomatose. So I'll pass around this example of a foliose lichen over here. And so those, if the stalks produce apothecia, they can be red or brown, and that is one of those reproductive structures. The crustose lichens, those are the microlichens. Like I said, they're very thin. They're actually embedded within the substrate, so they lack that lower cortex. And so, you need to use a rock hammer if it's in rock in order to remove the substrate. And one reason why it's kind of out of scope for today is that you do need to have a compound microscope that you, with identification, it helps to look at the spores. One other example of, or form of crustose lichens are crustose leprose lichens, which are the dust lichens. And those ones, there's an example of it here. They're kind of just these loose composites of fungal hyphae and serratia, which we'll cover. And you can actually run your finger over it, and it looks like dust. Here's kind of a close-up of it. They actually can't derive water directly from rain. They have to get water directly from the moisture in the air. So here are your typical reproductive structures. You have your apothecium, which some of them lack rims. Sometimes the color can be the same as the vegetative body or different. So that kind of helps in identification. You have your isidia, which is on that lung lichen, which the serratia and isidia you can kind of think of as starter lichens, but isidia are kind of have a better startup than the serratia because they actually have part of the cortex with them. Since it has part of the cortex or skin, they can sometimes look shiny. And they can maybe have different colors on the tip of them. They can also be branched and do some other funky things which help with ID. Otherwise your serratia, sometimes they can be look really powdery, or they can look really mealy, like cornmeal. They can have different looks to them which help with identification. And the serratia, it's just the algae with the fungal partner. It doesn't have the cortex with it. If you see patches of serratia, they are called serralia, and sometimes those serralia can have different shapes to them, whether they're saddle shaped or barrel shaped, and other forms to them. So the serratia, they're the powdery clumps of the fungi and the photobion. It's the smallest reproductive fragment. Like I mentioned, the patches of them are called serralia, which they can have different shapes, the stripes, balls, etc. The location of them and texture matters for the identification. Here's a close-up of a hooded tube lichen, where the thallus actually upturns, and it's on the underside of the lobe margin, and we have a really good example of that here. The hooded tube lichen, it's also has this inflated look, like it's kind of filled up with air like a balloon, and you'll be able to see that on this. So I'll pass that around over here. And I have another example I'll start to pass on your side of the room here. A smaller version. But feel free to take these out of cases and look at them under the hand lens. And then you can look at, compared with the acidia that's on the lung lichen, but lung lichens can have both acidia and serratia. So the acidia, they're starter lichens that kind of just have a better head start. They can have different shapes to them. The location matters, and some of them can be black tipped, and some of them can also have the branching. And there's a lot of things too, depending how deep you get into this, there are things that can be described as acidia-like, and you just kind of have to learn to tell the difference. Your apothecia, usually saucer or cup-shaped, but that's not always the case, containing those spores. And like Sue mentioned, this is the form of sexual reproduction. Those spores are stored in these little sacs called asci. And this apothecia don't always, have to look like the red or brown you might have seen on such like an illustration of British soldier, which we'll cover. They can have different forms. Here's an example of it too on a dog lichen, which someone thought kind of looked like a canine tooth. So they can have different forms, and they can also take different forms like lyrae, which we'll look at an example of that shortly too. As I mentioned before, risings are not the only way of attachment. Sometimes you have these thick mats of fungal hairs. Top or bottom can be described as tomentos. Some of our foliose lichens, like your rock tribes, can be described as umbilicus. Exactly the way in which you think the word is, kind of has a central point of attachment, a holdfast in the center of it. And those holdfasts are, attachment disc or surface that allows it to stick to the substrate. Texture matters with lichens. They can have sometimes look grooved or channeled, caniculate, or as you saw in the lung lichens, sometimes they're reticulated. You can also have these cilia-like projections from the lichen. You might read something that it has pustules, which are kind of just like warty projections of the cortex. Whether the lichen is smooth or wrinkled or looking prunose matters. Here's warts on the common toadskin lichen, those being the papule or the pustules. If something's described as pruna, a powdery look, it kind of looks like it has an icy coating to it, that's actually calcium oxalate that is being produced that protects it. There are also different breaks in the cortex that allows for a gas exchange, and these are helpful with identification of certain genera, such as with your punctilia. This can help you determine that one genus. Maculae are kind of just these pale spots or splotches in the upper cortex, which way I did think of it is just it's a thinning of the cortex, just where the cortex gets very thin. Whereas there's also something called pseudocephalae, which are actually more pore-like, looks like pores in the upper or lower cortex, which don't have any thalline rims to them. So I mentioned earlier chemical spot testing. That's the only way sometimes to tell species apart. If you are a researcher, most people determine this through liquid gas chromatography, but sure enough, I left that in my garage, so we can't do that today. No, those machines are very expensive and sophisticated, so it's out of the scope for most people. but we can do color spot tests. Some of those things you'll see in the literature, just these little letter abbreviations, whether it's positive or negative. One is your typical household bleach, C, which helps with it. Potassium hydroxide, which is described as K. You can get potassium pellets and dilute them, or you can actually buy the KOH solution from medical supply stores, which they're relatively inexpensive, and you can find it on Amazon as well. Something that is a little bit more difficult to obtain and also potentially hazardous is your paraphenylene diamine, described as P or PD. You can usually only buy it in bulk, and it's one of those things, just like anthrax or something like that, that you have a lot of it, and it's bad stuff. So you just have to just be smart about it. There hasn't really been anyone that's used it. Most people don't. The Brodo and them in that book described they've used it for 40 plus years, never had any issues, but it does cause staining, so you do have to watch that. What you do is you put a small drop of it on the cortex or skin and look for a color change. Does it change yellow? Does it change red? Does it do a color change from yellow to red? That sort of thing. Or do you expose the medulla? So one of the things that you also might want to have in the field is a pocket knife, or if you're working under a dissecting scope, a razor blade, and you scrape the cortex layer away, and also that algal layer away to expose the medulla. And that can also have different color changes. You can't just do the cortex, oh, that changes yellow. The medulla might actually do something different, where it turns bright red, pink, that sort of thing. Sometimes you'll see in the literature you need to do a KC test. So you actually put a drop of your KOH followed quickly by a drop of the bleach, and that also will do a different color change. The chemical spot tests reveal different acids that the lichen produces. And like I said, it could go from yellow to red. Some of the yellow can vary in color. Some of them are described as a gold color. The one to kind of notice is for P plus red, grayanic acid, that's named after Reverend Fred Gray, which was a lichen collector in the early 20th century that we have extensive lichen collection for him in the WVU herbarium. So that's kind of cool that that's named in his honor. Something else that's kind of cool is sometimes exposure of the medulla to UV light can also help indicate a species, whether it changes blue, white, or has orange fluorescence. And here's an example of Cladonia grayi, which fluoresces pale blue whenever the medulla is exposed. A couple of notes here: if you don't want to go by KOH, your house lye can also, in many cases, provide the same kind of chemical spot test result. If you get the Brodo book, you'll also see in there that they do some chemical testing using your Lugol solution, which you can also get that from different medical stores, but that is a helpful spot test too sometimes. And another one you might notice, which I haven't personally used, is alkaline iodine, which might turn blue or violet for acetic acid. Lichen substrates, the preference for it can help identify the species. The lichen derives very little from the substrate other than a place for it to grow. Some of them you'll find more in conifer versus deciduous. The degree of decay of dead wood can make a difference. Rock, there's a huge difference because of the ability to retain moisture more for longer periods of time in sandstone. There's chemistry that varies on how those rocks are binded together. And then when it comes for limestone, you know, it's drier, and there's also a varying degree of the calcium carbonate present. Some lichen, yep.
I was going to ask, are the substrates ever affected by the growth of lichens? So when you see a tree absolutely covered in lichens, does that harm the tree at all?
Well, I know as far as like on rocks and stuff like that, because they're kind of producing those acidic compounds that it's actually over time breaking down to rock.
Right.
Yeah, as far as on a tree goes, do you know that, Sue, if it's the same thing?
Well, basically, I think the weathering overwhelms the effect of the lichen on a rock. You know, it's more like the freeze dry is going to freeze and
Yeah.
Freeze and expand. It's going to have a more profound effect than the lichen. But the net effect overall tends to be positive because they hold. Yeah, they hold water and nutrients. Both mosses and lichens are nutrient and water traps, and studies have shown that that's beneficial to nutrient cycle in the ecosystem.
That makes sense.
Yeah.
So, but yeah, that's a great question. And so it seems like too that, you know, the ecological community benefits then from actually having those lichens present,
Yeah.
or more wholesome ecosystem. Sometimes lichens will actually be on top of other lichens. You will see that a lot with your rock tripe. Sometimes you'll see lichens growing on top of them. And also other bryophytes can be a potential substrate. Moving into our species profiles, we'll see how much we can cover of this within the next 25 minutes or so before giving you guys a break and then starting mosses. This one is, I try to focus on ones that you can find around the park, the pipestem, very easily. There's the Ramalina americana, which to me it kind of looks like chewed leather. You can find it in any open wood trail or woodside here, found on tree bark. The thallus varies a lot, sometimes being narrow to wide. It kind of looks flat. The thing that varies a lot is the level of striation. Sometimes it's very smooth, sometimes it can look striated, and in live specimens, the upper and lower cortex can look very different from each other. You won't find any serratia on this, and the apothecia, they are on the lobe tips and actually have a pinkish, creamy look to them. So these are the apothecia here. And this is a fruticose lichen, so there's no risings present. So what I try to do with this too, if you want to not just get the bulleted points, but learn more about these lichens, I provide the page numbers for these guides, and you can write down more notes from that species profile in the book. Yeah. Oh, okay. We will cover one like that. That's part of the crustose lichens. We'll cover one that's called the pink earth lichen. That is exactly what you described. It looks like just a little cotton candy. Yeah, it's very cute looking. If a lichen can be cute, so we'll cover that one shortly. But I have a couple examples of the Ramalina americana, which I'll pass around for you. And again, you feel free to take these out of the bags and play with them. The one that's in the bag here actually has the fruiting structure. The other two I don't think actually has the fruiting structure present. But I'll just kind of put a couple of these in the room, and you can just see the variability in the growth form. But basically, if you just look on the ground on any of these trails, you are bound to find this somewhere where it fell off a tree and it's just kind of hanging out on the ground. Ramalina americana. There's also one that grows on rocks that's Ramalina intermedia. But this is the only one that grows on bark.
Can you make that up one more time?
Yeah.
you.
I also want to try to cover certain generic, because my goal here today is for you to pick up on, you know, the color, texture, variation. If you can kind of narrow it into a certain genus, then I did my job. One of these that's really cool are the usneas, your beard lichens, which it kind of looks like the tree is growing a beard. They vary typically from pale green to yellow, but you'll find there's some like Usnea rubicunda that the cortex can actually have a red tint to it. They usually have these short fibril projections coming off of it. The surface can be described as papillate, again kind of just these little projections of the cortex, a serratinate or a citiate. The branches can be rounded to angular in cross-section, but what makes this unique is that it has a dense central cord that's very elastic-like. It's really cool. And so I do recommend finding one and taking a knife or something, exposing that central cord and looking at that. That medulla can be white or reddish. And the taxonomy, at least according to Don Flanagan, is a little bit in question. And I've noticed too with some of the specimens that I worked with in the herbarium that there seemed to be a lot of variability. So potentially there are other species, but I guess we'll see. Did you have a question?
Could you explain the reagent reaction? Is that, doesn't react to potassium and chlorine, but does to the combination?
Okay, yes, yeah, that's a great question. So for, there's 3 West Virginia species, and all of them will test like this, where the skin is K negative and C negative. So negative to KOH, negative to bleach. And if you do a drop of the KOH followed by a drop of the bleach, though, then you actually will get a reaction. So the 2 chemicals separately, no reaction, but when together, they do have a reaction. And I have 3 different species examples with me today to go ahead and pass around. Let's see here. And you'll notice too that they vary in color. Let's see here. Which one? Can't tell offhand which is which. The small one in the case is the Usnea subfloridana, but we also have today with us Usnea strigosa and also Usnea. Okay, this is the other one in the case is the Usnea rubicunda, because you'll notice that there's a reddish tint to it, and also it's not blackened at the base where it attaches. So I'll pass these 3 different forms around. But these can get really big and prominent. These are just really small examples. But if you look closely on any of the trails out here today or tomorrow, you'll definitely see these guys. One of my most favorite is the Usnea strigosa whenever the apothecia are present. Relatively speaking, they seem to be in higher elevational areas. Found on deciduous trees, and the thing that's you notice right offhand is that it has all these fibril projections coming off of it, including around the apothecia, which to me it's the beard lichen that looks like a Venus flytrap. So that's what helps me. It doesn't have any serratia or accidia, and the medulla actually it can vary from white to red. And we'll hopefully today see this guy on, I know there's at least one somewhere on the trail that has 2 fruiting bodies that you can try to get a good picture and look at it. The rock tripes that I mentioned earlier, those are the ones that has that umbilicus attachment to it. They are vary from medium to large, but they can form these large maxon boulders. This was taken, I think, somewhere out by the Heritage Point. And if you go to places like Cooper's Rock State Forest, you'll just see these boulders just covered in them. They kind of have a leathery look to them. They're smooth to pitted, and some of them have that powdery coating on them, which helps distinguish species. Lower surface, pale brown to black. They can have these plate-like or grill structures on the bottom of them. And the apothecia are in these sunken pits on the front of them. There are 3 species in West Virginia, but the one that you are most likely to encounter that's the most common is the Umbilicaria mammalulata. Smooth rock tribe, yeah. Thank you, Sue.
So, Matt.
Yeah.
So the rock tribes are edible.
I think I've heard, no?
There is one that's used in Japanese cuisine that's highly edible, but for the most part, they contain acids that make good dyes.
Right, yeah.
You're better off, it's been said, oiling your boots.
Yeah, so probably not recommended, but yeah, the cool thing that the Native Americans used to do is that both for your rock tribes and for your toad skin is that if you soak it in ammonia over a long time, that you actually get a really cool purple dye from it. Yeah. The next one that you can also find in the same areas are Lasalia papulosa, your common toad skin, in these in rocky areas. It has button-like apothecia on the front, so you can see the apothecia look a little bit different. And the dimples below match, basically where you see the blisters on the top, the dimples below match the placement of those. And here you can kind of see a more active version of it than the picture I showed earlier in the presentation. I do have some really small versions of it that I can go ahead and pass around, as long as I can find it. So it looks like I have at least one version of it I can start passing around. And this kind of helps show a side-by-side comparison. The lichens pose for me. So you have your rock tribe on the left, kind of has a brown olive look to it. It's lacerated. But on the right-hand side, it's pretty clear that you see those pustules, or on the top, those wart-like projections of the common toad skin. Another one you might encounter, the species varies whether it's a wet area or a dry area, are your sticklebacks. They're, in this case, it's your common sticklebacks, also known as a leather lichen. You find these at dry rock outcrops. If it's a wet area, then it might be Dermatococcum liridium, and the lower surface differs, or the lower surface is white. These, to me, they kind of look like potato chips. No acidia or serratia. They are actually dotted with something called perithecia, which, if you get into lichens, you'll have to be able to identify those as well. And they're basically these like flask-like structures within the lichen that contain fungal, that contain spores. Moving on to your common dog lichen. Even if you don't go out today on the trail, if you take the access, the paved access road down to the lake about halfway and just look at the left-hand side of the bank, it is just covered with dog lichens. There are these large mats that are really beautiful. The common dog lichen you usually find in shady areas. Again, no serratia or acidia. The top is fuzzy, lobes wide, and the bottom have these tufted risings. And they have unique apothecia on the lobe margins that some that might look like to some dog canines, or at least are saddle-shaped. And I do have some examples of this today too to pass around. But I definitely recommend seeing the lichen in its full form as opposed to just a part of it and really get appreciation for it. And this is a picture of it, not just a part of it. You can see that darker color to it, indicating the cyanobacterial partner. Moving on to probably one of the most common ones you may have encountered and not realized it is your Flavoparmelia caparata, your common green shield lichen. To me, it kind of looks like chewed bubble gum. The look to it, these yellow-green rounded lobes. They can. There's a lot of variability to this one, just like your Ramalina americana, in terms of how smooth or wrinkled it is. There's no cilia projections or pseudocephalae pores in the surface. They have these granular-like serratia, and the lower surface is usually darker, but it could be browner on the edges, and it has this margin with no risings on the bottom of it. And I do have an example of that too. If you go over to the Arboretum right next to the lodge, the trees are just plastered with green shield lichen, so you'd be able to see some really nice versions of it. There is another form that only grows on rock, which is Flavoparmelia baltimorensis, and in that case, it has dense insidia on it, and it's only found on rock. This one, though, you can find on rock too, right?
Yes.
Yeah. Yeah, so that you have to make sure that you're looking at serratia or insidia. So here's an example, kind of showing the rock version that has the extensive insidia on it. And another one that is really common within the park here is your yellow ribbon lichen, which it really has these really nice kind of ruffled margins where all the serratia are concentrated. And you can also notice a color difference with them too. They kind of seem to be a little bit more of a bright green. But the look of it is very distinct. I do have a couple versions of that as well, and you can kind of see a difference in the color as well. Lichens have a lot of variability in their looks depending on the amount of shade that they're in.
The ribbon lichen kind of like a flat version of the British soldiers, does it look? Most of the same coloration like the British soldiers?
Well, we'll show the British soldier, and it'll be a little bit different. The British soldier, you'll...
I know they're tall, but it seems like a similar color.
Oh, no, it's okay. Yeah, the coloring with the British soldier can vary a lot. But yeah, but it's also like a yellow-green color. Do you know the color of why it's...
Okay, basically the colors of lichens will fade tremendously as they dry out. So for varium specimen of British soldier, they actually lose its bright red color. So it's best to see them live.
Right.
As we will be doing.
Yeah, so you'll see in this guide that some of the pictures I took are from herbarium specimens, so the color might be a lot more faded and not as vibrant as what you would see if you were actually out in the field. And on one of these two, you might see some splotches where the color change varies a little bit, and that's where I was doing some chemical testing on it. Another one you'll see out here is your Melacroa, your axle bristle lichen. You can find these also in similar habitats, but it has a bluish gray color to it as opposed to yellow-green with narrow lobes. And you will find some simple cilia in the low axils, but you won't see, they won't be as obvious as like some of the ones we'll be showing later. The lower surface is black with simple risings. The medulla is usually white, but it can be yellowish sometimes around the serratia patches. And there's also some, this is one of the ones that, at least for me, it really helps to do some chemical testing. And so there's a couple different things that you can test for there. I don't know. Okay, moving on to another group. These are also throughout the park, your ruffle lichens. And if you go up to a lichen that looks ruffled and has all these cilia-like projections, it's a good starting point to look at your Parmetria. They also have a broad naked margin underneath. Kind of maybe hard to see in this picture, but you don't see any of it here. The cilia, like I said, they're kind of present on the margins. And differences in pseudocephalae and cracking and such that helps make a difference between this punctilia and some other genera. One of the more common ones is your powdered ruffle lichen, having a powdered look to it because of the presence of the serratia up there. Kind of a pale greenish gray, the cilia long and abundant. The maculae in it, the thinning of the cortex can vary. I do have one example of that I'll pass around, and even without a hand lens, you can definitely see the cilia right away. This is another one that is easy to recognize, your hammered shield lichen. It does have the hammered look to it that jewelry or a ring might have. Only found on bark. You have serratia present on the cracks and ridges. The risings are helpful in this case. We've been seeing a lot of lichens that have simple risings, but in this case, they are right angled. And there's also the presence of the pseudocephalae or distinct pores that show through the cortex on the upper surface. Okay, so here's a hammered shield lichen that's growing next to what? Pop quiz. What? Beard? I heard beard, yeah. So that's your beard lichen there, your usneus. Punctilia, speckled shield lichens, they have pseudocephalae. Lower surface varies from pale to black. They have unbranched risings to the low edge, and this is one of the cases that it can be helpful to do the chemical testing, see if the cortex is K+ yellow. And there's another genus, Flavopunctilia, but it will be more yellowish in color. I do have an example of Punctilia rudecta, which we are going to cover next. Find it on both bark and rocks. And it's called rough because of the densicidia on it, which are sometimes branched. And you see pseudocephalae, especially at the tips, where you can kind of see this paler coloration off the back. The hooded tube lichen is one of the ones that I mentioned earlier. Is kind of looking and has an inflated look to it, like it's filled with air. which the way it looks can vary, but it typically fans out at the tips. And it has those puffy lobes where you see the Soredia present. And I think I do have already those specimens out. One I wanted to cover just because it can colonize in large mats on trees is Physea milligrana, your millie rosette lichen. Common, found in every county in West Virginia on bark. And the Soredia on it can be very heavy, which is described as mealy in some of the textbooks. The lobes overlap and they look crumbly, not only from the Soredia at a distance because there's a lot of fine cuts in the lobes. And this is just an example of, close-up example of just a large mat of them, and then if you back up on a tree, you would just see the tree is just covered in it. And I do have one Physea example with me. It's not that one, but where is it? I have an example of Physea stellaris with me, and it is it gets its name Physea stellaris, the star rosette lichen, because it kind of has a growth form that looks like a star, the way in which it grows. And there is a lot of apophisia on this too, which will give you a really nice example of what that looks like. So I'll pass that around. But this one's also common to your Physea stellaris, star rosette lichen. And when I did a chemical testing on it, the cortex was K+, medulla K-. A really cool one is the lemon lichen, which is also known as the candle flame lichen. It gets its name concolor because the rim of the apophysia matches the thallus color, which, let's see if you can see it on here. It's kind of blurred out, but there's one up there.
Now, would that one typically have yeast in it because of its color?
That's a great question. I don't know if that's actually produced from the main fungus or from the yeast. Would you know?
Oh, they've only found the yeast in a very limited number of lichens.
Okay.
So that probably hasn't been tested yet, the yeast.
So I guess TBD.
Yeah.
Probably not.
The cool thing is that you find these, basically if you know where some birds are nesting, you're likely to find this lichen growing underneath the nest if it's a nest that's used, or nest site that's used from year to year, because they like to grow in nitrogen-enriched areas, so where those bird droppings are. You can find this on bark, occasionally rock. It's really small. But if you see something really small in yellow, just take a close look and see if you see finely divided lobes on it, and it has these aggregates of Soredia. If you do not see finely divided lobes, then it might actually be a different genus called the I'm having a brain fart.
Oh, Italy, or Candelaria.
Yeah. Yeah, there's another one that does not have the finely divided lobes. So you can find thick mats of reindeer lichens out here in some parts of the state park. One of these are the star-tipped reindeer lichen. Typically high elevation areas, and there's something called pycnidia on the tips of the branches, which are globular flash-shaped structures that produce conidia, which are asexual spores. The amount of branching makes a difference with reindeer lichens. The branchlets are typically in fours or groups of two, And one of the things, great features where they start to reindeer lichen too, is you kind of look for what they call a open axle, an opening there wherever the branching occurs. Another example of the Cladonia scalaris that will pass around. But next I'll cover the most common reindeer lichen that you'll likely encounter. Well, this is kind of show its growth form here. This kind of looks like these, almost from a distance, mats of cauliflower that's organized on the ground. The one that's a little bit more common is your Dixie reindeer lichen. You can find it among mosses and soil. Has these wide dichotomous forking. The branching does not appear windswept in one direction. It's in different kinds of directions. If you see one that looks windswept, then it's actually probably your rangy farina. This is kind of shown what it looks like there, and there is a lot of this at the park. So I'll put this on this side of the room, the Cladonia subtenuis.
Got some moss growing within there.
Yeah, there's yeah, yes, exactly. Okay, and I'm going to try to get through the rest of these real quick, next 5 minutes, so you guys got a break. So one of the ones you see that's textbook lichen is your British soldier. What's unique about this one? This is the species that was used to determine that lichen relationships, that it's more of a controlled parasitism, that they use this species to determine that. You can find it on soil, rotting, decaying substrates. No acidia or serratia present. These bright red, lumpy apothecia, they're really Kodak friendly. The podia are typically unbranched. K and C by themselves are negative, but you can't get a KC gold. And it is incredible the amount of variation with these. You just need to see enough of them in the field, because each time when I first started out, I'm like, oh, this is a different one. Oh, this is a different one. But it's British soldier, just it can look a lot different depending on the amount of shade. And that's that specimen that I passed around earlier. That was the mosaic of 4 different species. British soldier was present on that one, and this kind of shows it being present among several other species too. And I have a couple examples of that one to pass around. Let's see what I have left to make sure you guys.
Mack, I have a quick question.
Yeah.
How do you, you put them in these little boxes, how long do they stay good for?
Lichens are great for collection. I typically store them in just brown paper bags. Get a collection, put it in the freezer for a couple days just to kill off any insects that might be living on it, and then just store it in a dry place, dark place, and the colors will fade over time, but I mean they're great for keeping. They don't have to really do anything special for them. Yep. As far as I understand, as long as they have all the right conditions, they'll just keep on growing. But granted, they grow very slowly each year. And so if I'm ever collecting, I usually just do it opportunistically, unless there's a large mat present. I usually just look for stuff that, wind or like a deer put its hoof in and brought up some of the soil, or if a tree branch fell, and I'll just take that. So I usually try not to collect a lichen unless it's already just loose, or a lot of it present. But yeah, as far as I know, they just keep on growing. But you'll see too that if you go out in the field today, we do show how the growth forms, where they kind of like on a foliose lichen, you'll see these concentric rings sometimes, and that's just because the way it grows, that the inside of it kind of died off, and the new growth is on the outside of the ring. So sometimes you can see these concentric rings, and that's just because of the way it's growing. You'll see the old and new growth. And I want to try to cover at least one more before giving you guys a break before the next talk, just because it's rather unique. We'll look at some crustose lichens out in the field, but this one is, I wanted to show you one hybrid growth form, which is called crustose fruticose. It's your nipple lichen, but it also goes by the name gnome fingers, which I really like. It's the only species in its genus in North America. And if you go down on the Dent Tree Trail, if later today, part on one of the where it opens up and there's exposed soil, there are large mats of this. The thallus is granular, and the secondary thallus are these irregular, inflated, hollow stalks. So it kind of reminds you of the hooded tube lichen and having that, inflated look. But I have some examples of that I want to pass around and for you to enjoy too, because I think it's just really cool looking. But this is an example that you can't always put a lichen in the box. It kind of does what it wants, and there's some intermediate growth forms. And with that, and just because we did talk about it earlier, one of the crustose lichens is your pink earth lichen, which you find in disturbed soils. That's present at the park as well. The apothecia are these little cute, kind of cotton candy pink apothecia with these ribbed stalks. And I do have a really good example of it that you can see without even using a hand lens. And kind of for comparison too, just to show how these podia can vary so differently, I also have a Cladonia here called a ladder lichen, which you could just get a difference for how these podia can look really different. So I'm going to start with the ladder lichen over here, which is just really cool to see. And with that, that's all I can cover today. For today, I want to make sure that you guys have enough time to take a 5-minute break before Sue starts. And we'll get to, I put this cartoon up here because we'll actually get to see today, if we go outside, the competition for substrate space with lichens and mosses. Some really cool competitive stuff going on. Yes?
You said that they use these like that. You consider insecticides? I still check for insecticides and things like that that are out in the environment. It's likely that they use that to check for that type of substance.
Well, I know that they bioaccumulate heavy metals, but yeah, as far as other kind of compounds that you might find in insecticides and stuff like that, I'm not sure how that might be uptaked. Right.
I don't know how much detail they look into laying out here.
Right. Yeah, that's a great question. I'm not sure. But thank you for your time, guys. We're going to take just a quick break, and there's other specimens up here.
Are you serious? That is brilliant.
No problem.
problem. There's not a better reason to go home.
You can take it.
That's good to know. I know students use it. Oh yeah, take one of those. Take this.
It looks like the sun might be breaking.
Yeah, these I have to take.
But it's raining. I'm seeing rain.
Is it still raining?
Yes, it is.
Yeah. So remember, there's free Ohio gods up here if you want one.
You know, it's really kind of disturbing. We're in West Virginia. Give Ohio stuff away.
I
But you
know.
won't give us the West Virginia stuff.
Tell the DNR they need to put that back in French funding.
Absolutely.
Okay, no problem. It's a great book.
I guess those are for later, maybe, they can take out. And I'm so excited, actually, that
Oh yeah, it's just like there's so much diversity in your own backyard. You don't have to go far. And plus, you know, just like usually have firefights around the same area, so you can stay at one rock for hours. You know.
You can also watch it just like this, but he's got
Yes.
Yeah, that's right. There's no way to
Yeah, and I've been telling people they need to return it, so don't worry.
That's right. You got it.
Yep,
We got to recycle these things.
well, somebody will enjoy your life. Yeah, sorry, so I'm trying to
I've got it.
yeah, they can stay there.
Oh, yeah. Yeah, you can.
So there are some.
Yeah.
So like in old college days, I spent a year studying like this, and it's like, oh my God, this is so cool. This is well, not just little.
I love these. Yeah, Mack. Frantz. Oh yes. Yeah, email me and remind me, and yeah, because they cover. I try to cover all the unique ones and common ones.
Thank you so much. It's all really confusing.
Oh,
But it's good.
it's good. It's all.
So do you think these are available?
Not with our office, but you can get them if there's officers in the problem. If you contact them, you can probably get it from that organization. Ohio Loss and Life Association. They probably have.
In the insurance. Yeah. In the lab.
What's the scientific name? Does it say? It's not going to tell me. I'm guessing Rainier Springs. Rangers.
Yeah, it is.
That might be Studlar.
Stellaris, okay, I'll look it up.
Yeah. Yeah, Ranger Farina, that's only if they're all facing one direction.
Oh, interesting.
Yeah.
You said this was Stellaris?
I think that's more Studlar's, yeah. Cladonia Studlar's. Yeah.
Thank you.
Well, I'm going to be an orthopedic, so that would
Do you need anything else?
Oh, well, I need to, I want to see if it's ready now. I'll need to, yeah, somebody should
Oh, I need to get back to
to get it back on the screen. Just from the beginning.
Yeah, I would just hit escape and then go back to the beginning. Yeah. Easiest way.
Yeah, nice job.
Oh, thank you. Thanks for correcting me with the
Oh, well, that was
prophecy.
just
I was
one of those things.
nervous.
Yeah, no, that can happen. Oh, do you want to turn this off?
Yes. Yeah, when I was going through it, I'm like, well, it's
I just throw it out in their backyard.
So can you,
And they weren't interested in spraying on the grass, but it was never experienced. They don't want to take it. His dad was have you seen the church?
No, yes.
Yeah, because it was about 80% free there, 70 or 80%. But, you know, really curious guys and the women.
This is one of the something
My friends just slow down there. They have black flowers. And they said that they said that as soon as they got really that part of it, go right into bread. Just
I would stop and say it's one like this. There's smoky eye, there's policy in that. This is what you're okay to the first time. It's such a cool one, you'll see how that was surrounded by this like ribbon. It's not sure like so I started with
It looks way better than what I had, so.
it's a starting point.
Exactly.
Which is good.
Sing it now. It's stuck. Did that come on? It'll stick right back on.
Testing, 1, 2, 3. Can you hear me?
It doesn't. Well,
Certainly, yes, that's great. Very good. Yeah, how's it felt, Tigger? There's a number of species. Yeah, if you check the lichen book, you'll see. It's a really neat genus.
Yeah.
But it's easy to recognize.
Because it does look like a pellet.
Yes, it does. Yeah.
Thank you.
Okay. Card? Oh, I brought all I had. You can get those free from the West Virginia Indiana. I mean, not West Virginia, Ohio. All you have to do is write them. That's where I got them. So I got a mass of them.
Yeah, they were up there. She also said write them also. And I saw them, you could get them online. But I didn't go up there. Down there. I know.
So, I know. Yeah, thank
We did ask her to put it on.
Oh, it's good to get one of these booklets. There were some more up here.
Val.
Yeah, this is the Ohio.
They have some.
All right, are we ready for graduates? Hey, I am Sue Studlar. I was at West Virginia University for 23 years teaching botany and currying mosses and lichens in the WVU herbarium. Supervised a number of independent study students, and Mack was actually my last student at WVU. Great pleasure to work with. We had a great time in the field yesterday. We spent 3 hours in the rain going on the trail and came up with, here there are 20 bryophyte species under the scopes here that you can look at some point. You just need to remove the lid and turn on the lights, and they will dry out. You just leave the lids off, but be sure you remove the lids so you really see them in all their beauty. I'd be happy to be back here. For 11 years I did moss walk at Blackwater Falls State Park for the DNR. Now I reside in Asheville, North Carolina, having retired from WVU after 23 years. But I'm happy to note I will be teaching bryophytes and lichens at the University of North Carolina in Asheville next spring, and I lead walks in the Asheville region. So I'm doing the same kind of stuff, except sprinkled with the wonderful cultural activities for which Asheville is famous. It's full of plays and music and hoppy ales, so it's really a great place to live. But I'm happy to be back here as well. So I'm here to share with you the joy of moss. This is absolutely wonderful weather for mosses and lichens. So you just have to kind of get in that mindset. I have a feeling we will be lucky this afternoon. So we shall see. But I think we will be. It looks a little brighter out.
It does.
So like Mack, I'm trying to give you kind of an overview of diversity of mosses with emphasis on the growth forms, so you learn how to look at them. So the first question is, it took great minds centuries to sort out the differences between mosses and lichens. So if you're confused, you're in the company of greats like Aristotle and Linnaeus, so you don't need to feel bad about it. So what do you think in these slides here? Does anybody have any ideas on which of these would not be mosses? After Mack's talk, I bet you can pick out a couple of them. Anybody want to choose a couple?
2.
Number 2, right. And what is that?
5.
And number 5, very good. Oh, see, Mack, you did a great job.
And number 1.
And what is number 1, do you think?
It looks like some sort of
Looks like
lichen.
It's actually, that's a tricky one, that's Spanish moss. It's actually a pineapple in terms of the family.
Oh,
There's a couple more fakes. Any ideas?
7?
Number 3. I put that in deliberately because it doesn't look like a moss. It's Pacific Northwest rainforest, and it looks like a lichen, but it's actually a moss. But, so I'm kind of hoping you'd fall for that. Any others you see that look confusing?
7.
This actually is a bryophyte. Mosses, we use the term moss to refer to bryophytes in general, but they're actually mosses and liverworts. And the liverworts include those that look a bit more like lichens. Those are called the thalloid liverworts, and that's what that is there. So that is actually a liverwort. A couple more fakes in there. Any ideas? What would number 6 be? Any ideas? Believe it or not, that is a fern. But that is the cryptic phase of the fern life cycle. And that looks very much like a liverwort. All right, there's one more.
4.
Number 4, and what would that be?
A club moss.
Yes, this is a lycopodium. It's been renamed Huperzia. It's a primitive lycopodium. These are the sporangia here. Okay, so there's the list. And by the way, I'll have my computer over there so you can go through if there's the opportunity, you can go through the PowerPoint on your own also. Okay, now, so how do you define a bryophyte? These are the 3 great groups of bryophytes: the mosses, the liverworts, and the hornworts. What do they have in common? Can you see anything there that they all share?
Spores.
They have that sporophyte that is unlike that in any other group. That is basically there's a single sporangium on a stalk. And each of those would develop from a fertilized egg. So technically they are said to be monosporangiate. And basically that is the definitive feature. You can never go wrong if you see a sporophyte of that type, although you can see they're somewhat different in the 3 groups, and we can talk about that more as we go along, but just notice there that they look quite different. So the green phase is the gametophyte phase. The sporophyte is always attached to the green gametophyte. It will never be on its own. It has no rhizoids, which are root-like hairs in bryophytes. So having an attached, dependent, stalked sporophyte with one sporangium, that is really what defines a bryophyte. So the definitive feature is based on life cycle. Just about any other criterion you can get in trouble with, but this will always work. So in reality, bryophytes, unfortunately, often lack these definitive sporophytes. Any ideas on why? Pardon? That would be that. Yes, actually, that's often the case. That's a good point, because if you want to see the spore capsules, you want to go into a habitat where there's enough light. So often along trails, the bryophytes will be fruiting abundantly, and you go into the deep woods and you have more trouble finding them. But basically, the most important problem, probably, or equally important, is that they are typically unisexual. There are male bryophytes and there are female bryophytes, and they tend to become extremely widely separated. They may be on different rocks, or they may be on different continents. And if the males and the females are separated, there is a great difficulty in bringing about fertilization, getting a zygote, and from that getting the definitive sporophyte. It so happens that moss sperm are very weak swimmers, get tired after a couple centimeters, so the sexes have to be very close together. So basically, considering this problem, some species have evolved what are called splash cups. And these are among the most beautiful features of mosses. These splash cups are designed by evolution to splash out the sperm so they can go more than a couple centimeters. They might go 10 centimeters. Or in rare cases, it might even go a meter. So you can see there's still a problem. If you're on different rocks, you're going to have, the male is going to have trouble finding the female. So these are often called male flowers, just kind of symbolically. They look attractive to us. Of course, the female moss couldn't really care less about the beauty of the male moss. It's something more that catches our eye. It's a purely functional relationship between the sexes, or chemical attractants can be involved as well. But this is one of the more spectacular features of bryophytes. Unfortunately, have those problems, and then additionally, males are often rare. And this is population bryology is a hot research topic in graduate schools now, and one of the questions is, where are the missing males? They're just not out there. They're scarce. So you have a lot of bryophytes that do not have these definitive spore capsules. But still, you have a lot of males that bloom beautifully anyway, even though it's, I suppose you could say in a sense, futile. But they're certainly beautiful to us. I don't know. Yes.
Is there asexual?
Oh, absolutely, and I'll show some of that as well. But back when I was in school, we read the same poems my parents did, and I remember one line from Thomas Gray's Elegy in a Country Churchyard. Of Fulmenia flower is born to blush unseen and waste its sweetness on the desert air. You could say something similar for the male bryophytes. Okay, so this is something you learn to look for in spring. In some habitats, you can see these beautiful male mosses. These are all male mosses. You can see they look different in the different species. This is a species you'll see, saber-tooth moss. We'll see some haircat mosses. They have beautiful male flowers, and there's several species. And we will see the slender star moss, which has much smaller male flowers, so to speak. So in bryophytes without the male flowers, the males are there, but it's more subtle. So here we have one of those thalloid liverworts that looks like a strap and might be confused with a lichen if you hadn't got such a nice introduction to lichens. But now you probably wouldn't mistake that for a lichen. It just doesn't look right, does it? The color is off. It's too green. Lichens are mostly fungus, so they will tend to be paler. But you can see these little flaps on the thalli. That's where the sperm sacs or the antheridia are in the ribbon work. It's hidden behind the ruffles. And in the peat moss, you can learn to recognize male branches. And in the peat mosses are very round sacs. So the inset there shows a round sac of another common species of peat moss. So how are you going to recognize the females if they do not have these showy flowers? you can recognize them by their beautiful offspring. So that's what you're seeing there, the spore capsules, and look at the differences in the appearance of the spore capsules. They're all beautiful, but they are differ in orientation and shape and the persistence of little protective caps. We will certainly see an unbelievable amount of the brocade moths, Hyptum imponens. That is really flourishing out there on the trail that we're going. So you should all learn that one readily. And it has capsules that are straight, quite different appearance from the beautiful rose moss with its curved capsules, or the haircap moss, which has hairy caps. So the amazing thing about the hairy caps, it's kind of hard to believe, but they're actually transformed egg sacs. It's just one of these amazing developmental features where you go from a sac holding an egg to a mass of hairs that is over the developing sporophyte. The sporophyte starts out as a needle or a spear, and we'll see these out in the field. And then with the protective structure of the mother, the transformed archegonium or egg sac, the offspring develops. And if you pull it off too soon, the offspring will be, the capsule will be abnormal in many species. So it's a great example of maternal control of the development of offspring. Can you imagine putting a little cap on your child that controlled how it developed? And if the cap stayed on long enough, you got a beautiful child. If you took it off too soon, not so good. It's an interesting concept. Okay, so here's some more beautiful offspring. These are the needle or spear bryophytes I was talking about. Technically, they're called seedy, and the cap there is called a calyptra. Capsule is the capsule. Notice the inclined sporophytes there on the second species, and that we can call a lid or operculum. So here you can see in different species 3 stages of development, all of which we can readily see in the field today, and I'm confident we'll be able to go out there. You've got the spears with the calyptra on top, the mature moss sporophyte with the operculum, which protects the developing spores, and then once they're mature, the lid falls off, and there are these wonderful teeth at the mouth of the capsule that help disperse the spores. So you can see young, mature, and old capsules. We looked hard on the road bank and finally found one patch of what just looks like algae on the bank that had several sporophytes of the pogonatum. So we will be able to show that to you because we marked that little patch with flagging. So hopefully we should be able to relocate that. But that's kind of an interesting moss, pogonatum. It looks like algae, which is what a juvenile moss plant looks like. It looks like algae. But so this is actually a species that is sexually precocious, like some salamanders are sexually precocious, whereas really the larval stage is sexually mature. See, would that include the hellbender, or is that a mature salamander?
Just the mud puppy.
Pardon?
Just the mud puppy.
The mud puppy would qualify as a larva that's sexually mature. I know some salamanders do that as well. Okay, so more on sporophyte diversity. We were delighted to find in the field yesterday the nut moss. Now this may look pretty different from the others I've shown you, and we've got that actually under the scope here. It's a beautiful little moss. Once you can learn to recognize it, you'll spot it readily on eroding banks and so on. It looks like little nuts scattered over the bank. Those are the capsules, and they just have extremely short stalks, practically non-existent. And I have seen rain fall on these. When the conditions are just right, you have rain and sun. This happened once at the West Virginia Botanic Garden, where I've also led walks for years. My assistant held the little colony up. The rain struck the nut moths, and these little puffs came out. It was just extraordinary, but I can't promise that. that was just luck. We will also see a lot of the rock tuft moths, and you can see more readily the hairs on the protective cap from the mother. They're called the calyptra, and just to the right of that, you can see a lid right up there, not quite tall enough, just coming off. So you lose the calyptra, then you lose the lid, and then you get the spores gradually being released from the capsule. Mosses look incredibly different, wet and dry. This is what one of the very common mosses out here just around the lodge that we see, the crispy tuft moths. You can recognize a good part because of the way it dries all crispy, and then when it gets rained on, it expands very quickly and looks like a different species. But notice the capsules, again, showing the diversity. The stalks are pretty short, so it's immersed, but they're not as short as in the nut moths, and the form of the capsule is really quite different. So you will find these virtually stalkless or sessile and short stalk capsules often are on rocks and tree trunks. Can you think of any evolutionary reason for dispensing with a stalk if you're elevated on a trunk or a rock? I mean, do you really need a stalk? The wind will just blow the spores right out. So this is a common evolutionary trend. Okay, so as I mentioned, the calyptra falls off, then the operculum, and then the spores are released. So this is prickly grimmia, which really is transformed when dry versus wet. If you go by the New River and you see what just looks like black crust on the rocks, you're not sure if it's a moss or not, but you look closely, you see little bits of green. That's probably a moss in the Gribiaceae or black rock family, and you can see the difference in it wet or dry. Just it is absolutely transformed. They make the rocks really beautiful right after rain, and then they look just very black and dry between rains. Now here you can see the lid of a grimmia being shed, and this is what the teeth of the grimmia look like that surround the opening of the capsule. These teeth are really marvels because they are actually the remnants of cell walls, and they respond to moisture changes. So they go in and out, in and out. I remember watching, I mean, you could just be hypnotized by this. I was watching this under a stereoscope one time while listening to Bob Brandenburg concerto, and it's just really a trip. I recommend it. And we have some mosses with teeth here, and actually you might see some movement. You can easily induce movement actually by putting a little drop of water nearby, and they will respond to the humidity change. So that's one of the fascinating features of bryophytes, having these teeth. Why bother releasing them a little bit at a time? Why not just get rid of them all at once? You think of
Greater success of time lapse between release of.
Absolutely. So you're just increasing your chances of the spore landing somewhere that's favorable. You know, different wind conditions, different circumstances. So generally, moss capsules do not release their spores all at once. It's usually a gradual phenomenon. And one of the treats of the black rock mosses is that the teeth are bright red. So you poke your head into one of those black mats, add a little water, and you might be rewarded with a mouth with bright red teeth looking right at you. Okay, so then these spores will develop into the gamete plants, and of course the gamete plants produce sperm and egg, and from the fertilized eggs the sporophytes develop. So that's the moss life cycle. And that's really important because that is really the definitive feature of mosses. Okay, so now let's look at some of the outer groups. This is, I don't know, well, one of my former students, and I've got Joe Rohr, the second author in this wonderful field guide to mosses. Their team made up common names for mosses which were much better than the ones I made up for the DNR. So I've largely switched to those of my former student. That's what you want. You want your students to do better than you. So I was really pleased to see these common names. They're really very good. But it does not include the liverworts. The British have standardized liverwort common names, but they're not that meaningful to American ears, I don't think. So this particular liverwort, which we saw yesterday and we may run into again, and I have it under the scope. They call the Grove earwort. I don't know, kind of strange. It might be easier to just call it Scapania, like the common Scapania. And here you can see the sporophyte bursting out of the mother. This is what's called a precocious sporophyte. Rather than having the needles that gradually mature with the maternal cap controlling everything, the sporophyte develops completely inside an envelope of the mother, and then it bursts forth. It's kind of like a traumatic childbirth. Just boom, out comes the offspring, and the stalk, everything's there. The stalk just has to elongate, and the capsule is pushed out into the air, and then it splits open into valves and releases the spores. And there's these little wiggly filaments that move in response to moisture that help to disperse the spores, which often have already germinated. So liverworts sporophytes are really strikingly different from those of mosses. You can also see they just look different. What do you notice about the leaf shape of the liverwort compared to the mosses we've seen so far? The plant is flat. That's characteristic. Many mosses are flat, but liverworts are almost always flat, and the leaf shape is almost never pointed. Instead, it's round. You see a round, flat bryophyte. They love water and shade, and especially if you see what looks like a massive spaghetti with dark balls on the end, it's a liverwort. What will happen is it'll release the spores and then collapse. So you might see a rotten log covered with what looks like spaghetti, and that's a liverwort. And there is a couple of mosses that also split in valves, but that's a very unusual and primitive feature in mosses. So that's more of a cliff moss. So precocious sporophytes, capsules open by valve, and I should have said almost unique. There are actually a couple other mosses that have valves, but it's really unusual. So it would probably be unique in your experience. All right, now moving on from the life cycle, one of the amazing features of bryophytes is that they're essentially resurrection plants. Lichens behave like slow-growing plants, and they basically have resurrection features too. And the scientific term for that is cricotohydry. So here is starburst moss, which you will definitely see. And on the left here, you can see how the leaves have all gotten really contorted. Different moss species have have different ways of drying out. And that's one of the fun things to note in the field, and it really helps in identification. With the atriums, they just get kind of all curled up and tangled, and it takes a while. They're sort of slow at it, and they revive more slowly than, say, a Grimmia. So when they're dry, they shut down. So it's pretty amazing the way they can revive, often in just minutes. So basically, this is, you know how it is. Your kids have pets, and then they grow up and leave, and you may leave you with a pet. This is one of our sons, who's now a ranger. His last surviving pet, Meet Bud, the Argentinian horned frog. Only in the last 2 years, he's 24 years old now, has he hibernated near the glass so I could actually see him and see that he moved. I was worried about him as if he were a strange child. You know, what's he doing buried for 6 months? Isn't he getting stiff? But we could see that he moved a little bit during this long dormancy. And he basically got up recently, just a few days ago, and since then he's eaten 2 worms. He hasn't really regained his normal appetite yet. And after waiting for 6 months, he emerged, and then the next thing he buried himself. I was furious, you know. It's time for you to get up, you lazy toad. But so I mean, this is kind of an amazing parallel, really, to think of an animal being able to shut down like that for months. But do you think it would be exactly parallel? I mean, the mosses actually lose water and become extremely dry when they are dormant. They're great at taking up water and also at losing it. And that's key to their dormancy. As it turns out, Bud is like in a little space capsule. He's like in a little sack of mucilage, so that he is protected from drying out. And he sits in our bay window for 6 months and is quite I guess, content in his own way. But I think well, amphibians are parallel to bryophytes also in that they require water for the sperm to get to the egg. So bryophytes are often called the amphibians of the plant world, but this is another parallel that might be less familiar. So what do you think? Is Bud pretty cute? I don't know. My husband thinks it's very strange that I pull out my phone and show pictures of Bud rather than of our son.
How long can they live?
They're supposed to live no more than 10 years in captivity, and he's 24. So I guess he likes it there. Okay, so lichens, bryophytes have growth forms which you can look at as beautiful compromises. There are stresses for bryophytes. For one thing, where they live is always a temporary habitat. You know, you see a log there. It might be there for 20 years. It's certainly not going to be there forever. They're on a slower, like plants in general, they're on a slower time scale, and that disappearance of a log or a tree over the long haul is a problem. So that's a problem. The primary stress for bryophytes in nature is wet heat. And they can tolerate just about anything when they're dry, but if they're wet, their metabolism is not equipped to handle that. So that's why you won't see many bryophytes in habitats where you have the combination of wetness and heat. It's very stressful for them. Also, being suffocated by litter is a big problem. I mean, they have a particular life strategy that involves being low to the ground, and they cannot grow very tall. Very few bryophytes are competitive with the higher plants, or vascular plants they're sometimes called. Tracheophytes is really a better term. So they can readily be overgrown by vascular plants. And they also have the stress, as Mack mentioned, of competition with lichens and also with other bryophytes. Now, all solutions in nature reflecting evolution are imperfect. So one strategy would be to escape to new habitats by way of either the sexual spores or the asexual propagules. What would you think would be the chances of a given spore actually surviving, becoming a new moss? I think it'd be good or poor, or extremely poor. So, it's compromised, but at least there's a chance. Likewise with the asexual propagules. And basically when conditions are unfavorable, they shut down. But if it's wet heat, they can't shut down. So that's a problem. And then they can grow opportunistically throughout the year. And then of course they can occupy severe habitats that rise above the litter, which is not severe for bryophytes, but may be severe for tracheophytes. And then there is the general strategy of poikilohydry and the specialized growth forms. There's the distinctive niches that different species will show, but basically there are problems of scale. I mean, bryophytes are not just little vascular plants. They are really very different in their life cycle and in their metabolism. And poikilohydry is a strategy that works great for small organisms, not so great for large organisms. So bryophytes and lichens are poikilohydric. Tracheophytes or vascular plants are not. Some of them approach it, but it's never quite the same as with a bryophyte. And of course, the amphibious lifestyle that is connected with their way of life is very limiting because they can't have sexual reproduction unless there's free water available. So, this is wonderful bryophyte web. They can grow, they can have sex, they can, just thrive in these rainy conditions, and they will look their best. I mean, if you don't feel you're looking your best out in the rain, just think of the bryophytes. They look great, and you can kind of transfer your identity to them and maybe feel better. Okay, so basically, moss gardens celebrate growth forms and diversity of mosses. Basically, you don't need to know all the moss species to appreciate them. Like lichens, they have these very recognizable growth forms and the differences in the sporophytes and the gametophyites, and you can kind of just appreciate the texture and color differences and be open to their particular kind of beauty without having to know all the names. And this is one reason they're celebrated in moss gardens. It's an ancient tradition in Japan. There's an active tradition in Germany, and I was privileged to see a moss garden, which they use their old pools for aquatic plants for mosses. It worked very well. North Carolina has a famous, let's see, here we go. A woman who's relentlessly campaigning for mosses all the time. That's what she does. She goes around the state and does workshops on moss gardening. This is Moss and Annie, very gregarious advocate of moss gardening, and she sells mosses. She grows them on, if you can believe this, the non-skid underlayer of carpets. That's what she grows mosses on. And then she can cut whatever size piece of moss carpet you want. And she can't understand why not everybody can grow mosses as beautifully as she does, but she lives in the wettest part of North Carolina. Get 100 inches of rain a year or something like that. But she's a moss enthusiast and has a website, and this is her book, Moss Gardening, which you might enjoy looking at. This was just one little small moss garden in Japan, and actually that was just a little atricum there, a starburst moss. They use some of the very common species that we will see to great effect in Japan, creating miniature landscapes, taking advantage of the different growth forms. So the Berlin moss garden included something we will see in great abundance, the white cushion moss or leucobryum. And you may have noticed there was a net over the garden. Any idea why?
Something wants to eat it.
Pardon?
Something wants to eat it.
Oh, well, actually, it's what's under the mosses. The birds are always flipping mosses. Because, you know, they're anchored typically with these rhizoids, which are just little hairs. And a bird can flip the moss out of the way and get the grubs that are underneath. So they have all their mosses covered with netting to avoid their beautiful gardens from being destroyed by birds. But notice how distinctive that growth form is. It's a perfect little hemispherical cushion. It's called either the white cushion moss or the pin cushion moss. And it is white when it's dry, green when it's wet. There are two species. We will see, we'll probably see both. I've got one of them under the scope here. There's a large one and there's a little one in the inner grade, but they are distinct. This is an unusual moss in that the leaves are effectively several cells thick rather than mainly one cell thick, because the leaves are basically mostly midrib, and most of the cells are dead and empty and store water and air. So when it dries, it looks white. So you can readily learn to recognize this moss. I'm sure you will today if you don't know it already. In Cacapen State Park, there are beautiful, extensive white cushion mosses. Have some of you been to Cacapen? And have you seen the white mosses on the ridge? You might notice it now if you hadn't before. So it's something like a cactus. And it's basically, this is one of the mosses, because it looks beautiful but different, wet and dry. It's very often a feature in moss gardens. And the small one is often abundantly fruiting. The large one is more likely to shed its leaves, and the leaves become asexual propagules. Now, this was something I saw at the West Virginia Botanic Garden that I found just amazing. This is a short turf moss. These are ones that form a turf-like pile on a carpet. It's called short if it's less than 2 centimeters. And this is one of the common species, Dicranum montanum, which if you look at under the microscope, it's nothing but a bunch of little babies. You know, you saw starter lichens. These are mostly starter mosses, and that's basically what it is. It's just lots and lots of little mosses that can easily start over and produce more mosses. And you will see this very commonly on tree trunks, just a brilliant green. Now, I was going over the route for a walk at the Botanic Garden, and I saw a cloud arising from the tree trunk on which this Dicranum montanum was growing. Any ideas on what's happening there? There's a cloud coming from the tree.
Is it releasing spores?
Pardon?
Releasing spores? Releasing spores.
Say that again?
Releasing its spores?
Oh no, actually that's a good suggestion. I wasn't understanding it because I know what the answer I'm looking for. All right. What is it releasing? They're poikilohydric.
Water or steam.
Water. Yeah, you can actually get clouds coming from trees. Now that you know about this, you might see it. It was so striking. It reminded me of what we saw looking out the window of our room this morning, where we see the fog rising from the canopy, and we now know that climate is in good part controlled by the plants beneath. You cut down the trees, you can actually affect the amount of rainfall you get per year. As has been known for a long time, founder of plant geography, Alexander von Humboldt, noted that in the 1800s. So this is a miniature of the pipestem landscape here. And who knows, if the sun comes out, we might even see something like that. It's something to watch for.
Is that water coming off the moss? Or is
Yes,
that just...
it's coming right out of the
Because I
moss.
see that a lot of mornings at my house.
when the sun starts to shine, when it comes up, all the trees are just like exuding moisture.
So you can see that
I've seen
also.
it. Oh, a lot. Yeah.
Oh, really? No, this is the first time I've noticed it. But in this case, the trunk is just cloaked with moss. And what those mosses do. Yes. I mean, the bark can hold some moisture, but the outer layer of bark is cork. And by the way, that's why the lichens are hurting the trees. They're anchored in the cork. The cork is dead waterproof cells. The lichens are not getting into the sugar and water conducting tubes. So it may very well be the moss, unless you have a really spongy old tree, but if you do, it's probably covered with moss. So yeah, this is a really neat phenomenon here. So this is a short turf. And the common name for this is crispy broom moss. We will see a lot of dicraniums, and I've got some dicraniums in the dish, and their leaves tend to be sickle-shaped, and it looks like they're blown in the wind. And like an old broom that, you know, you've been using a little bit too long. And so this one becomes extremely crispy when it's dry. So this is one of the great names used by my former student, crispy broom moss. Better than the one I used, which was the same as the British Biological Society used, which was mountain fork moss. Mountain, and then the teeth have fork, but you normally don't see the teeth, so, you know, crispy broom moss is really a much better name. So I've been happy to adopt that. Okay, so I have been at Pipestem before. In 2002, the DNR asked me to look for the rare moss, the Ammons twist moss. This is a specimen that's really tiny, maybe 1 or 2 millimeters across, and it was named in honor of Nell Ammons. It's a very skinny book. I guess I accidentally left it in the room, but she wrote the Liverworts of West Virginia, which is still very useful. Well, basically, I'll be very busy with my teaching. The only time I had free was Thanksgiving weekend, and we drove up to meet the wildlife biologist, Jeff Henke, who was going with me, and it was snowing. And, you know, we were supposed to climb on these steep cliffs by the river, but it was the only time I had. Well, the next day, the sun came out, just as it's going to come out for us, and you could see everything, even the tiniest little moss under the ledges. So I was able to find in Pipestem State Park Ammons twist moss. And just last year, the DNR botanists found it again. they kept looking for it, but when it's dry, it just looks like dirt. You cannot see it when it's dry. It's impossible. It's tiny, and it's crisp, and it grows in the dirt, so it looks like dirt. But apparently from what the botanist P.J. Harmon told me, they went out under similar conditions, and they found it in several sites. So it is definitely been verified as here, Ammons twist moss. And so it's a good example of the problems of working with mosses. They change so radically, wet and dry, and they can be very small. And this is an old herbarium specimen, 1974, and you can see what look like little tiny leaves in the center of the rosette, and those are gemmy or leaf-like asexual propagules. It can reproduce that way, and also by fragmentation. Bryophytes, like lichens, are great at reproducing by fragments. In the case of lichens, of course, you need both partners, and you get both partners in a fragment. In the case of bryophytes, just about any piece of the bryophyte can grow more bryophyte. That's true of the gametophyte, not the sporophyte. So this is one of these examples of extreme separation. Females are in scattered places, and there are no males known. So no sporophytes have been described for this species, but it's living on, and in its own little niche. So that is Syntrichia. And this is another Centrichia that I put in to give you an idea of what it would look like fresh. This is the fragile twist moss. You see in the inset there some leaves. When you separate the leaves and look at them under the microscope, they just fall apart. And each little fragment there could produce a new moss. But you can get an idea of what the leaves look like. They're clear cells at the bottom that are specialized for water absorption, then green cells up above, and this strange kind of pale green color. Not enough to be a lichen, but basically that's an effect you get on some mosses like leucorrhizome because of dead, empty cells that reflect the light. In the case of many of these tiny little mosses that grow on limestone, the leaf cells are covered with little bumps that